It's still sharing, yes?
Yeah, you're good.
Yeah, right. So let's go. This is basically the application of machine learning in biotech, especially the genomics sector. And yeah, also great thanks to Deep for helping me out throughout this. So we had a few meetings, he helped me out. It was cool. Then the flow of our talk will be going through the genomics prerequisites, then the machine learning, going through the limitations, as well as what my ideas are, where I'm going with this presentation basically, and then the summary. So with the genomics prerequisites, first we have DNA, of course, when we talk about genomics. So DNA is nothing but deoxyribonucleic acid. It's just a really long molecule, basically, or many molecules joined together. And even in that, what we can drill down into is that when we see the structure of DNA, it is double helical. That is, two separate strands are wound together in a double helical structure. Further, what we can also see is that we see these special molecules at the end here. These special molecules, what they do is on each of these strands, they go up to the center and then they bind with the other strand. So the molecules that start from the end go till the center are called nucleotides. So those are nucleotides. And in those, the central part, the central gray parts are the four bases. So bases are basically the side chains of the nucleotides, which can be classified into these four molecules. So this is the chemistry of it, basically. So the four molecules are adenine, cytosine, thymine, and guanine, ACTG. And these are basically present in all cells and contained in chromosomes, such as 23 pairs of chromosomes in humans. And yeah, different parts of the DNA are contained in different chromosomes, basically a wrapping mechanism for the DNA. Because if we stretch out the DNA, it becomes too long to be contained into the cells. So chromosomes are the wrapping mechanism. And each DNA contains genes. So if we think of DNA as a long sentence, genes are the small phrases of it that carry meaning. And when I say meaning in the case of genomics, it's basically have some profound effect upon expression. So those are genes, and yeah, that's the DNA as a whole. Then we get to the DNA replication mechanism or DNA copying. This will be helpful to us when we get to the further parts of how genomics or gene editing is done. This is basically what happens is a protein gets bound to the DNA right here, which is initially double helical. This protein is helicase, which unwinds the two DNA strands, and polymerase gets tacked onto each of these strands, and then produces the other strand based on rules. And these rules are nothing but the fact that these bases occur as complementary pairs. So yeah, they occur as complementary pairs, which we can see in this diagram. A only goes with T and C only goes with G, and so on. So this process we can see in more detail in this video right here.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
So yeah, that was basically DNA replication, and what we want to take away from that is replication isn't exact, and it many a times causes mutations in the DNA. So especially on the complementary strand, we have the fact that the DNA is replicated discontinuously, and that basically causes more amount of mutations in the DNA, which further causes diseases when those genes are expressed, or rather due to the mutations are not expressed. Then moving on, we have the whole process of how DNA gets translated into proteins and how it affects our body, basically. So we have the double-stranded DNA again. The process is basically first carried out by the transcription part. So transcription is turning the DNA into single-stranded RNA. And the difference between these two is that the T, which is thymine, gets converted into a uracil molecule denoted by U. And those are the basic differences between DNA and RNA, other than the fact that RNA is single-stranded. RNA can further be translated into proteins, and the proteins are the ones which carry out the different functions in our body, and that includes basically all the functions. In the previous slides, when we saw the enzymes ligase ligase or helicase or even polymerase, those are all proteins carrying out basically the replication process itself. So it's a cyclic process in our body. And here we can also see another fact that a group of 3 RNA molecules or bases gets translated into one single protein. And yeah, that's constant throughout. So we'll look into that more now. So those are basically called codons, those group of 3 molecules together. So codons can be translated into different proteins. So if we say that the previous process was analogous to compiling code, where the code got compiled into a binary file, basically. This was, let's say, keywords of the code, right? So if we have AUG molecule, it denotes methionine, a protein, which is specifically the starting molecule for the replication process or the translation process. This is where translation for the most part starts in proteins. And when I say most parts, it's because the process is imperfect again. And again, similarly, we have the stop codons, UAA or UAG even, or even UGA. These are all stop codons, whereas all the other codons translate to different proteins. Now, again, these different proteins are different molecules and they have different chemical properties. And based on these different chemical properties, they carry out different functions. This is the list of all the translated proteins. Again, for the most part, there are some different proteins present in different species. Again, some outliers. And yeah, we have some exceptions in that case. But for the most part, these are the different amino acids. Yeah, so these can be called as amino acids specifically. And when they combine together to form a longer chain, those are more scientifically called proteins. All of these can be denoted by 3 letters, or more commonly even the single letter. This will be useful to us when we get to the machine learning part. And when translating RNA into RNA, so when transcribing DNA into RNA, or even when translating RNA into proteins, we have different reading frames. So the reading frame could start at point 1 or next over or further over. Depending on these reading frames also, we have different mutations that get induced into our DNA. And hence we find that more diseases come up as we age because more replication takes place within our cells and DNA gets replicated further. So we have 6 reading frames in total, the 5' to 3' strand or the 3' to 5' strand. And each of them have 3 reading frames in total. And then we get to specifically gene editing. But before that, if you all have any questions till now, again, just an intermittent. No? All right then.
I do have a question.
Okay.
What exactly is the problem here? Like, what are we solving? I feel
Right.
like if you give us
Right.
context, then we'll focus on the right parts of the biotech
Mhm.
that we should have questions about.
Okay, so the mutations are what we'll be focusing on. So the whole thing about machine learning and genomics is basically curing diseases if possible, and that takes us down different paths. So the different paths include directly correcting these mutations that occur. So that takes place through this process, that is genome editing, directly through CRISPR, which is one of the technologies here. And with that, in CRISPR specifically, what we do is create these other complementary strands, which are known as sgRNAs, which are bound to other proteins that get tacked onto our DNA and then edit out the mutated parts or the adversely mutated parts specifically. So yeah, and the other way around is directly going down to the protein structure, which AlphaFold is doing and which we'll be discussing. And AlphaFold is basically predicting the protein structure, and protein structures are important because they carry out the functions. So all the functions that proteins carry out are because of their structures, basically. So if we want to think of some molecule that's being taken into a cell, that's due to the receptor proteins. And
Pause you for 1 second there.
okay,
You've listed out a bunch of problems, and
all right.
we're going to get into all of those. My
Mhm.
question was much simpler than that. Like, is there one specific problem you're trying to solve, or is it just that you're giving us an overview of where ML is applied in genomics for a future discussion of like what you want to solve?
The latter.
Okay, got
An
it.
overview for
Got it.
future
Thank
discussion. Yeah. All right. So getting back to gene editing technologies, CRISPR is one of the more famous gene editing technologies that has come up recently. It stands for clustered regularly interspaced short palindromic repeats, means nothing right now. But it basically consists of this sgRNA part that we design as humans and then send it into our bodies to carry out some edits into our DNA. I'm sorry. Yeah, and those edits can basically then be used to eliminate diseases, cure other mutations, etc. And again, getting into the specifics, that's carried out through either gene knockout, gene knock-in, activation or inhibition, and screening. So when we say that, when we say knockout, it basically means just taking out a part of DNA, and that gets patched up by the mechanism that's already present in our cells. When we say gene knock-in, we specifically manipulate the replication mechanism to patch up a DNA in such a way that we get a specific mutation in place. And again, activation or inhibition depends on which parts we edit out. So if we edit out certain other parts of the DNA, it will activate or inhibit the expression of other parts of the DNA. Those are known as activators. Yeah,
So,
which, okay.
question for you mentioned a bunch of things with respect to genes. Could you, maybe I missed this, but could you again highlight what a gene is with respect to the DNA animation or the
Okay,
units that you explained so far?
sure, will do. So, all right. So let's say that this is a super long strand of DNA. A gene could be just this part. A gene is, yeah, a gene could be just this part from A to T. So a small part of the DNA which causes a particular function. So we have genes that cause breast cancer, BRCA1, BRCA2. Those are specific genes present on very specific chromosomes, which we have already found. Similarly, we have other genes, genes that cause sickle cell disease. So that may be at another part of the DNA. Suppose, let's say suppose let's say from here to here. So just specific parts of the DNA are the genes.
I see.
And
And so just to double check, like if the way you would find the breast cancer causing gene is compare the DNA samples of like a ton of breast cancer patients, probably across demographics and countries and whatever. And then compare like what are the subset of common genes that they have.
Right, right. So it was a long process. It was called the Human Genome Project, where they basically decoded all the genes that were useful to us in the human genome.
I see. That's very cool. Yeah.
Yeah.
I would encourage other people to ask dumb questions like this. It's totally okay. Like
Yeah, it
end of
is
the day,
because
something very useful. So yeah.
Again, in the machine learning part, you all will be more astute than me. So dumb questions right now will be appreciated. All right. Again, if any other questions are there, you all are free to ask. We come to gene activation or inhibition. So if we edit out some far away parts of the DNA, we can affect some other far away parts of the DNA from being expressed. And screening is basically locating some DNA, which can also be done with this sgRNA and Cas9 protein. We'll look into a video for this, so it gets sorted out.
The first step of a CRISPR-Cas9 experiment is to design a custom guide RNA that targets your gene of interest. This single guide RNA, or sgRNA, is an RNA molecule consisting of two parts, a constant region and a variable region. The constant region is the tracer RNA, also referred to as a scaffold sequence. This is a sequence that binds to the Cas9 protein. The variable part of the guide RNA is a cRNA, a 20 nucleotide sequence specific to your gene of interest. Designing an sgRNA can be done manually or with the help of online tools. When designing sgRNAs manually, the first step is to identify protospacer adjacent motifs, or PAMs, in your gene of interest. PAM sequences vary depending on the bacterial origin of Cas9. The most commonly used version of Cas9 is derived from Streptococcus pyogenes and recognizes the PAM sequence NGG, where N represents any nucleotide. After identifying a PAM sequence in your gene of interest, simply count 20 nucleotides upstream of, or 5' to the PAM. These 20 nucleotides make up your cRNA, which is then added to the tracer RNA sequence to construct the full sgRNA. The PAM sequence itself is not included in the cRNA, but note that it may be present on either strand. If your PAM is present on the antisense strand, be sure to count your 20 nucleotides in a direction that is still 5' to the PAM. Just remember to read the final sequence in the 5' to 3' direction. A PAM sequence, such as NGG, occurs every 8 to 12 base pairs in the human genome. Therefore, any given gene of interest has many potential PAMs. We recommend designing 4 to 5 sgRNAs per gene and empirically determining which one results in the most Cas9-mediated cleavage. There are several free online tools for designing sgRNAs. One advantage of using these tools is that they provide information regarding homologous sites elsewhere in the genome, which could be potentially targeted by your sgRNA sequence, resulting in off-target effects. One option is ChopChop, a target selection tool developed by researchers at Harvard. To use this program, begin by entering your target gene name or accession number, then enter the species. ChopChop will generate a list of potential cRNAs, their exact genomic location, exon strand, and GC content. It also ranks potential off-target effects elsewhere in the genome. As the technology for CRISPR-Cas9 gene editing rapidly evolves, we anticipate that the tools and recommendations for the design of sgRNAs will be continually improving. Through our own experience at Takara and CloneTech, we have identified some additional design tips. We have found that the best sgRNAs for several tested genes have a G at position 1 and an A or T at position 17. Please read the page below for more information on sgRNAs, including sgRNA production and screening. Designing the best sgRNA for every gene? That's good science. The first step of a CRISPR-Cas9 experiment.
All right, so yeah, that was gene editing. If there's any
Question
questions there.
before.
Yeah.
Okay, thanks. So okay, sorry. Question about that. So Like I followed parts of it, but like
Mm.
I didn't completely get the overview of why you need a fixed part and a variable part and what you're doing with it exactly again.
Okay, so let's get to this part again. All right, so this part right here is the Cas9 part, or we can say the tracer part. This needs to be bound to a protein. A protein, as we looked at before, is a molecule, and we need that protein to send this whole part into our body so that it binds to the our DNA. And then to bind to our DNA at a very specific location, we need this part, which is the sgRNA part, and it then needs to be around 20 base pairs long so that it binds properly. Once it binds properly, we have this protein right here, and this protein is again derived from bacteria, and this is basically, this whole process is derived from a bacteria defense mechanism against viruses. So what it does then internally is, once it binds to our DNA, it cuts around the complementary part that is present, and once it gets a double-stranded cut here, a double-stranded break, what our body does, our mechanism does, is tries to repair that cut. And then we can manipulate that mechanism to either produce a knockout, knock-in, or an activation or inhibition. So yeah, if we get into the details of that, there's whole process around it, and also how we can specifically get a knock-in for an edit or a knockout.
This is cool. So essentially you're saying is, if there is some existing DNA that has some sort of gene and you want to play around with it, what you give it is half of a new 20 units long strand of a new DNA, which will bind with this, and then some extra part of it that expresses some function that we need to be expressed. Is that?
Yeah, that's basically the gist of it. And then again, we do not need to care much about how we design this, because there's already existing tools for it. And yeah, you basically place an order depending on the gene you want to knock out or knock in.
And this is possible because we have sequenced every possible gene in the human genome.
Yes, but then its applications are limited, which we'll get to in the limitations part.
See. That is cool. That is useful.
Yeah, it is. Okay, so now we get to the machine learning part. You all know more about this now, but yes, I'll do my best. So let's do. So machine learning applications. Let's move this around. All right. So what applications do we have of machine learning in this field? So annotation of genomic sequence elements, even though we have annotated the genes already for the most part through a long and arduous process, the other parts which I mentioned, the other key parts such as enhancers, promoters, or as a whole they are called activators, those can be annotated with machine learning applications, of course. Then we have identify the disease biomarkers. Again, this might raise a question. If we've already done the human genome project, why do we need this? That's because there's limitations to what we've already done, and how diseases get affected, or how diseases get expressed in the human body. And then, yeah, we come to predicting proteins 3D structure from the amino acid sequence. This is done by AlphaFold. We'll get to it. Predict co-functional relationships between genes. So there's multiple instances where there isn't just one gene that's responsible for a particular function. There's multiple genes that are responsible for carrying out a function together. And even if one of them gets mutated, the function gets disrupted. So that's why we would need this, predicting co-functional relationships between genes, then predicting the gene expression, whether a gene will be expressed at all or not. This is because not all genes get expressed in this case means translated into proteins. So genes can be classified on a very surface level basis into introns and exons. So I know I'm throwing out a ton of terminology here, but exons are the ones which get translated, which is, contrary to what one would think, exons aren't excised really. They are the ones translated, and the introns are the ones which are not translated. So predicting whether a gene will be expressed or not. And then sgRNA effect prediction. So sgRNAs, which we looked at for CRISPR, their effects aren't perfect. They have off-target effects, on-target effects, and we do not want the off-target effects. So we want to predict the off-target effects as well. So I have a paper that we'll be looking at. Yeah. So yeah, coming to the annotation part is done by a GitHub repository that Deep referred to me. So this is ULM fit mechanism given by Jeremy Howard, if I'm not wrong. Yeah, and it's the genomic application of that. So, okay, I skipped a slide, I suppose. So yeah, this is the whole mechanism of it, how the structure looks. Like we have in ULM fit, we have the general domain language model, which is trained first. General domain in our case would be the whole DNA. Then we get to the task-specific model training. Task-specific model training, if we want to annotate or classify specific portions of the DNA, then we would use the training data of those specific portions of the DNA. So whether a promoter is present at a particular location or not. And when we get to the classification model, we change the head. Yeah, we change the attention head. I suppose I should have given the general structure first. So we have the embedding, we have the encoder, and we have the attention head in this case. And we go through the general domain training, then the task-specific language model training, and finally the classification model training. I apologize for that roundabout explanation here. But yeah. So we have this case. And finally, when we get to the classification model training, we change out the head part, and we do not carry over the weights from the previous attention head because the application has changed from going from a language model to a classification model. So prior to a classification model, what was being done in this model is that we were predicting k-mer portions of totally predicting the further k-mers of DNA. So yeah, so do you all want me to explain that more? I suppose a little more explanation would be required in that case.
I would ask some questions and get people to answer, or at least like acknowledge if they understood what you're saying so far, or if you're missing something, like get your audience on board.
Okay, so if you all have any
Sorry.
questions. Yeah.
Yeah. So I had. So you mentioned K-merge DNA, right?
Yeah.
So could you highlight what the actual target to classify or predict in that would be?
The actual. Okay. Yeah. We'll have to go way back for this. So again, we'll just be giving this type of data as an input. So the ACTG data, and in case of k-mers, if we have four k-mers, right? So it would be a group of four nucleotides that is given, 4 base pairs that is given to the model, and we predict the next 4 in that case. And again, we have parameters such as stride in this one, which they've optimized to get the best accuracy. So stride would be if we are going just 1 over, or if we are skipping over to the next k-mer directly from 1. Yeah, does that answer your question, or should I explain even further?
Yeah, so I think they consider the DNA sequence as a separate language and try to model around it.
Right.
Okay.
Right.
Okay.
Right. So yeah, the general domain training, the task specific training, and the classification model. Yeah, in general, that's that. They have the results here. So this really wasn't a published paper really, but their accuracy is good and beyond the other papers that exist. So yeah, they're getting good accuracy for classifying human promoters in this case. Again, if I have to give information about promoters, they are basically gene sequences, DNA sequences that are present close to a particular gene that control its expression. So yeah, basically that that much. Then further, we have enhancer classification. So enhancers are similar to promoters, although they are placed very far away from the actual gene that they control. And that's all the distinction is in this case. But yeah, they still control their expression. And we have, again, the classification results here between different species even. So
is there any specific reason why the results with the highest more, so like 5 entries at a time, didn't get anywhere, like any highest accuracy scores at any time? So like, is it something like if you take more entries in the model, then it cannot properly predict, or is there a grouping of DNA like 4 or 3 at a time?
well, I don't think they mentioned really. So I think they mentioned that around 3 to 4 worked best for them. I don't think they mentioned a reason, or if they did, I do not know. So we'll have to look into that. So yeah. But then heuristically, what should work is a 3-mer. So if we want, if we want it to be inferable, if we want the model to be intuitive really, then 3-mer makes more sense, because then we have the codons aligned with those. So, yeah, but that's not the point. The point would be to get better accuracy, and in this case, 3 and 4 is working. Yeah. All right.
So sorry.
Yeah.
So enhancer classification. So what exactly did it classify in the end?
Okay. So enhancers, to classify those, we would need heuristic knowledge. So again, some enhancers have been classified. Enhancers can be characterized by specific sequences. So again, when we get to DNA, that specific sequence could be ATG or similar to that. Yeah. So again, this is, we need a machine learning application for this, because DNA has imperfections, and it's not necessary that always an ATG would indicate the start of an enhancer part. So enhancers can be started by different DNA sequences, and for that we would need machine learning. Does that answer your question? I'm sorry, I'm losing touch with it right now.
No, it's fine. Yeah.
Yeah. All right, enhancer classification. Then we come to AlphaFold. This is interesting. AlphaFold is not actually taking the ACTG DNA sequence as its input. I think that's where we should start, because we've talked about that for the most part. What it's taking as an input is the protein sequence. So the protein sequence, amino acid sequence that we highlighted, the 21 amino acids with their corresponding 3-letter denotation and then the 1-letter denotation. It's taking that as an input, and then doing a lot of pre-processing on it in these 2 ways, and then sending it through these detailed plots, and there's a lot of trickery involved. So if we need to get into each of those parts, so let's get into MSA. MSA is nothing but multiple sequence alignment. I think so, yeah. So a single sequence, a single row of this can be thought of as the single letter representation of the amino acids in a series. And the output, by the way, is the 3D structure of the given amino acid sequence. So I hope we are clear about the inputs and the outputs in this case. All right, so then if we get to the single row of this MSA, it would be the single amino acid sequence denoted by single letters. And what MSA is basically tracking the co-evolutionary patterns of that particular protein sequence or the amino acid sequence. These amino acids have a charge associated with them, which is a positive or negative charge. And that's how basically, that's what dictates their structure. And further, that's what dictates their function, because structure dictates function. So now if we imagine that we have two amino acids in a particular vicinity, one's positively charged, the other one's negatively charged, they'll attract together and they'll come close to each other and bend the molecule that way. However, through some mutations, through an evolutionary process, if there's some mutation on the DNA level that causes a protein to change, and the protein changes significantly to a point where it changes its charge, such that they now have different structures, we would need another change in the DNA so that the function as a whole is maintained at the end, right? So if one protein changes its charge from positive to negative, that is, the protein as a whole changes, another should change its charge too, such that the structure is maintained and further the function is maintained. And that is basically tracked through MSA. So each row of this MSA indicates a different species or a different co-evolutionary sequence of proteins where where changes in proteins are observed, and we track those co-evolutionary changes basically. So one protein has changed similarly with other protein, and we track that, and that's given as the MSA representation. Was that fair? I hope it was clear. All right, and then we get to the second part, which is pairing. Pairing is basically what we have is we have the structure of some proteins already. So now what we do is get the protein sequence, the amino acid sequence over the columns, and amino acid sequence over the rows, and predict in the matrix the distance between any two amino acids. And that is the pair representation, which we get right here. And yeah, that's derived from the templates that we already have, structure database templates.
Can I mean, I should have asked this earlier, but like, what does having the 3D structure really give us?
Yeah, so this 3D structure gives us insight into the function of the proteins, and thus we can maybe heuristically decode the cure for different diseases, because just doing the CRISPR part, that is editing the gene on its own, isn't working so far, which we discuss in the limitations. So maybe if we get the structure of the proteins and we get to actually how the proteins work and what each protein is doing, the hope is that we can cure some of the diseases which we cannot cure just directly by changing the DNA.
Makes sense. Thank you.
Yeah. So then we give this MSA representation and pair representation to an Evo former. Evo former is kind of like a transformer. And this is actually, this AlphaFold 2 could take its own presentation as a whole, but what it does have is 48 different blots, which are not at all recurrent. Each of them have different weight initialization. And the MSA and pair representations are updated through each of those blots and passed forward. Within each blot, what also happens is that both of these representations exchange information. So if we have somewhat of a good updation in this one, it can affect the updation in the MSA representation too. Yeah, I hope it's fine. And what we get at the end is MSA representation and the pair representation. Again, the pair representation contains the distances within any two amino acids, and the MSA representation contains which two amino acids change together, that is, which two of them are co-functional, basically. Based on these two, what we can do is predict the torsion angles, which is done in this structure module. Yeah, so the structure module, what it does is predicts the angles. Torsion angles is, we have two angles, phi and psi, because we have 2 degrees of freedom in a linear molecule, right? So in a linear molecule, we have 2 degrees of freedom. Any molecule can be angled, any molecule can have 2 angles with the 2 adjacent molecules. And what this structure module then does is predicts the backbone, a backbone structure of the whole molecule. And yeah, I think, is this making sense at all? Because I hope I'm explaining it clearly enough.
So from what I understand, you have some pre-existing information already, which you want to incorporate into your prediction, which means when a new sequence comes in, the first thing you do, two parts. One is the sequence itself you analyze, and second is the structure of the combination of these individual elements in the sequence. You combine the combination of these elements or the structure into a representation matrix, and you also look at the specific sequences and figure out what they are similar to, and then you go ahead and then I guess it's like the EVO former and the structure module blocks, but
Right.
yeah.
Right. So within the structure module, what we have is it predicting the angles between any two atoms, and then we get the backbone of the atom. Furthermore, what we also have is that it basically performs a gradient descent upon the structure of the module. So when we get a prediction, it's again recycled back into the whole pipeline. And yeah, the structure is refined basically. In this, what we have an additional thing is that heuristically, even if we predict the angles, so even if the model predicts the angles, it's not necessary that those angles form a proper molecule. That's basically dependent on how triangles are formed. So if we consider three atoms in a series, so three atoms forming two angles with each other, and then we want them to form kind of a triangle in a case. So the distances between those atoms would decide whether the structure is actually viable or not. And this the structure module basically checks with that as well. And over time, it refines the structure and gets to a very high accuracy. Another feature of this is self-distillation. So very high confidence results get poured back into the data set and are used for training. So yeah, that. And what we have here, yeah, kind of a blurry picture, but what we have here is AlphaFold giving very good accuracy compared to its competitors, which was in the competition. It's a biennial competition, I suppose. And what we have on the y-axis is the error, which is the median root mean square distance. So root mean square distance would be the distance between the predicted atom's position and the actual ground truth atom's position. And AlphaFold gives a very good result with less than 1 angstrom of distance. So yeah, that was regarding AlphaFold in brief.
So what is G427? It seems to, is that the ground truth?
No, different models. So these are different models that were entered
So the
into
first
the competition.
one seems to have zero error, so I'm confused.
I think this was what AlphaFold was registered as. It's at least what I got. Okay. So, and then finally, we have this third paper, sgRNA effect prediction by Zhang et al. A fairly simple model. So if we are correctly predicting the on-target accuracy or the off-target accuracy, we have a label encoder. So sgRNA sequence. So sgRNA sequence would be supplied as an input here, which is the one we designed or one the computer designed. And yeah, it gets given as the input layer, produces embeddings. So this produces a vector, it produces embeddings, I suppose. Then it gets passed into a convolution layer. And yeah, a convolution layer, and then we have two different convolution layers which are identical and which both of them get passed into the attention block. And I suppose we get the predicted sgRNA activity at the end. So to be fair, I'm not thoroughly done with this model or this paper yet. So I'm not quite sure of what the results are being compared with. But yeah, the accuracy that they're getting is also really good. This is the sgRNA on-target prediction. So they have two models that they've defined in the paper. This is CRISPR on target, and it's in blue. Just good area under the curve accuracy. Yeah. And then finally, we have sgRNA effect prediction for off targets as well. So this is off target prediction. In this case, what we also give as the input is the actual DNA sequence. So to check if the effect is on any other place other than the target. Again, I'm not sure what the results, how the results are being evaluated on. I think it's the covariance that they are using, but again, not too sure of how they measure the effects, the actual effects. And again, the accuracy for that they've given in that paper is fairly good. This is a median interquartile plot, I guess, with again good accuracy compared to other models. Was that fair enough? Like we get to the limitations now, and then
hello?
Yeah.
So the CRISPR on target and off target is this same model, or was it that CRISPR
different models?
But
okay,
CRISPR,
so,
I know what you explained in CRISPR. So what was the name of that model that you explained right now in the chart? Because CRISPR seemed to be outperforming in both the things, right?
Yeah, so CRISPR on target, it outperforms the others in this case. These are two different models that they've defined in the same paper. One is for on target prediction, accuracy prediction, and one is to predict the off target activity. So this is for the off target, the structure for the model that predicts the off target activity. And yeah, in this case too, they have two different evaluation metrics here, which predict the off-target activity.
So the one that they proposed in the paper, which one of it is in this?
Blue. This is the blue ones.
So that's
the ones.
CRISPR, right? Okay.
Yeah.
Okay.
Off-target. All right, so now we get to the limitations, and then followed by where I'm going with this, or where I would like to take this as a project. So getting to the limitations. Only some diseases have a single gene correlation. So sickle cell anemia, I suppose, has a single cell correlation. So if we change that mutation, sickle cell anemia gene, to a correct gene, I suppose, it gets cured. Similarly, we have fairly promising results with breast cancer, although that's not completely cured, because that depends on two different genes, BRCA1 and BRCA2. However, for most of the other diseases, there's no single gene correlation. There's nothing that we can do to edit out a specific gene and say that the disease is cured. And yeah, it's been a big disappointment in the field ever since the Human Genome Project, since everyone expected all the diseases to be cured. Other part is that most diseases are caused not only due to multiple genes, but also environment plays a huge role. So if you get irradiated, you're going to get cancer, and that's not due to the mutations, that's solely occur due to the replication process. On the other hand, too, if you have a gene for tallness, you won't be tall unless you have a good nutritious diet. That is the environment playing into it. Yeah, and this effect takes place in the case of diseases too. And third is, it's not completely analogous to computer programming. So the initial tools I had was we could do a compiler kind of thing where we could directly get the sgRNA tracer proteins, tracer sequences, if we make changes into the DNA code. So that does not work because of multiple things, one of which, few of which are the effects above, but also there's a whole another article on it, on why we can't do that. So yeah, those were the limitations overall. So now where am I going with this? What we could do as project is, one was something that Deep suggested during our meetings, is this model which we had, annotation for genomic annotation. Since they are using ULM fit, we could use BERT, which I think is a viable viable choice. Since the data is readily available, it's just the DNA sequence data that we are using here in this case. That would go. Another thing would be actually predicting the disease, the disease biomarkers. So similar to annotation of genomic sequence elements, we could identify the disease biomarkers instead of just the genes that have been identified. Maybe there's other parts such as, again, the enhancers or the promoters that cause the genes to engage, the genes to get expressed, which cause diseases. Since, what we've discussed, it's not a single gene process, we could do that. So based on, again, data that we derive from correlations between who actually gets a particular disease, and then compare that with the DNA. So in that case, I think a viable choice could be type 2 diabetes, for example. So that's, again, not related to a particular gene as such, but it's a amalgamation of multiple genes that have been inherited through the generations. So that was something I was thinking of. So those were the basic two things I would.
One additional point. So for promoter, that promoter data, when I suggested using BORD, my main point was that using federated learning to train those models, because often we find that genomic data is very sensitive and it should not be leaked at any point. So we need BORD definitely to maybe improve on accuracy, but we could use those approaches to train them and see how it works. I mean, does it work well or not?
Right. Yeah. And federated learning is a good call on that one. However, what the movement that we've been going towards is, giving up the DNA data for research. And again, that's an ethical concern that's going on. So, Any other questions or thoughts? Because, that was it. So we went through the genomics, ML limitations, and then the problem statement. And
so
the
one thing,
references.
So one thing I'd add on that is you mentioned ethical concerns, right? So the whole genome project, still the data collection is going on in some parts. So they are now considering using federated, differential privacy for that, so that the user's data cannot be built back up and anonymity is not broken at any point.
Right.
So
so that was the presentation as a whole. So I suppose there aren't any other questions.
I would like, and I think you mentioned this in part, but I would also focus more, or at least in the follow-ups, I would focus now on like what problems are possible to solve without having massive compute. Given that we don't have access to massive compute. That being said, I saw something weird yesterday, which was like when I logged into NYU high performance computing, I saw AlphaFold 2 as like a model available. I don't know what that means, but I saw an option to start some sort of notebook with AlphaFold 2.
Their code is openly available.
No, the code is openly available. I was aware. I didn't realize they had integrated it into like our systems as something that you can use now off the shelf for a bunch of use cases. So I don't know if that opens up the possibility for me to prototype some of what you might want to do. But I just wanted to throw that out there, given that it's relevant to the discussion. That being said, again, like back to the original point was like, what sorts of problems can we carve out and attempt to solve? Or at least like what area would those fall into? Would those fall into, for example, like just fine tuning large language models on these kinds of things, like some sort of last layer training or something like that? Would it be some sort of inference task about protein functions given some sorts of input sequences? Anyway, you're the expert, but that's the direction I would want to hear more about also.
Okay. If we want an answer right now, I think the
You
part
take time, but yeah.
Yeah, but in short, what comes off the top of my head is predicting the protein function is highly dependent again on the structure, which is more compute heavy, as opposed to going with annotation or classification of certain regions of the DNA, which is training or fine tuning language models that already exist. So I think we would be going towards that at least.
I should
Also,
sorry,
Swapnil. Yeah. So regarding the compute issue, if someone has used ULM fit for annotating that kind of data, that means it does not require a lot of compute.
Yeah.
Because they might have had to train it from scratch because that it does not correspond to English, right? So maybe that could also help.
Yeah, that's true.
I was going to add a friend of mine called Meet Barot, who is at NYU, works on ML and biotech and specifically on predicting functions of proteins. So if there is more interest in this line of discussion, he's already on our Discord. So it's just a question of like asking him.
Oh, okay. That would certainly be helpful. I mean, I do want to go into this field as a whole, but again, the limitations are what I'm considering now. The fact that even big corporations haven't been able to make a dent in this field. So let's see. It would be certainly helpful to contact him and get his opinion on this. So you should do that. All right. So that was it, I guess. A long presentation, but
Awesome.
I hope I didn't lose you all.
That's good.